In continuation to the previous lecture, today we will start production of semi-synthetic penicillins. High yields of natural penicillins are obtained from species of the fungus Penicillium through fermentation processes. However, over the years, many microbial pathogens have become resistant to natural penicillins and are now only treatable with semi-synthetic derivatives. These are produced through cleavage of natural penicillin such that the G or V side chain is removed from the 6-amino penicillinic acid 6-APA nucleus of the molecule. Penicillin G or VG in presence of penicillin acylase is converted into V side chain and 6-APA. 6-APA means 6-amino penicillinic acid. Thereafter, by attachment of a chemically different side chain, a semi-synthetic penicillin product, example ampicillin and amoxicillin, can be formed. In addition, the 6-APA can undergo chemical ring expansion to yield 7-amino des-acetoxycephalosporinic acid, which can then be used to generate a number of important cephalosporin antibiotics. Example, cephalexin, cephalosporin, cefadroxil. Enzyme in analysis. Enzymes have a wide variety of uses in analytical procedures. Their specificity and potency allow both detection and amplification of a target analyte. Wet chemistry enzyme-based assays for the detection and quantification of a variety of substances including drugs are widespread. Enzymes also play a key role in immunodiagnostic, often being used as the agent to amplify the signal, for example in enzyme-linked immunosorbent assays. Within DNA fingerprinting technology, the enzyme DNA polymerase plays a key role in the amplification of DNA molecules in the polymerase chain reaction. However, wet chemistry analytical methods are increasingly being replaced by the use of biosensors, that is self-contained integrated devices which incorporate a biological recognition component, usually an immobilized enzyme and an electrochemical detector known as transducer. Biosensors. Much of the technological development of biosensors has been motivated by the need to measure blood glucose levels. In 2000, the World Health Organization estimated that over 170 million people have diabetes. Are predicted that this figure will rise to over 360 million by 2030. In view of this, many companies have made significant investment in R&D programs that have led to the availability of a wide variety of glucose biosensor devices. In 1962, Leland Clark Jr. coined the term enzyme electrode to describe a device in which a traditional electrode could be modified to respond to other material by the inclusion of a nearby enzyme layer. Clark's ideas became a commercially reality in 1975 with the successful launch of the Yellow Springs instrument Model 23A glucose analyzer. This device incorporated glucose oxidase together with a peroxide sensitive electrode to measure the hydrogen peroxide H2O2 produced during the following reactions. Glucose react with oxygen gives gluconic acid plus H2O2 hydrogen peroxide. In this device, the rate of hydrogen peroxide formation is a measure of the rate of the reaction which depends on the concentration of glucose in solution, thus allowing the latter to be estimated. As was discussed earlier in the enzyme catalyzed reaction, the relationship between the substrate concentration and reaction rate is not linear but hyperbolic, as described in Michaelis-Menten equation. This is also true for the glucose oxidase within a biosensor. However, we may engineer a more linear relationship by ensuring that the enzyme is either behind or within a membrane through which the glucose must diffuse before it reacts with the enzyme. This means that the system becomes diffusionally rather than kinetically limited, and the response is then more linearly related to the concentration of glucose in solution. Over the years, the YSI Model 23A glucose analyzer has been replaced by a range of much more advanced models. The current YSI Model 2900 series glucose analyzer is shown in the figure. This is a laboratory scale glucose analyzer. This instrument has a 96 sample rack that enables batches of samples to be run, with the analysis of each sample taking less than a minute. The instrument can measure the glucose content of a whole blood plasma or serum and requires only 10 microliter of a sample per analysis. The membrane-bound glucose oxidase typically only needs to be replaced every 3 weeks, thereby reducing the cost of analysis. These systems also offer advanced data handling and data storage facilities. In addition, these instruments can be modified to analyze a wide variety of other substances of biological interest simply by incorporating other oxidase enzymes into the membrane. To enable diabetic patients to take their own blood glucose measurements, small handheld biosensors have also been developed, which are in fact technologically more advanced because the enzyme and transducer are more intimately linked on the sensor surface. The first device of this type was launched in 1986 by Medicines and was based on technology developed in the UK at Cranfield and Oxford universities. The Exactech blood glucose meter was the size and shape of a pen and used disposable electrode strips. This device was followed by a credit card style meter in 1989. Such devices again rely on glucose oxidase as the biological component but do not measure the reaction rate via the production and detection of hydrogen peroxide. Instead, they rely on direct measurement of the rate of electron flow from glucose to the electrode surface. Basically, electrons are removed from the glucose molecules and passed via the enzyme to the ferrocene mediator, which then donates them to the working electrode surface, resulting in the generation of an electrical current that is directly proportional to the rate of oxidation of glucose and thus proportional to the glucose concentration in the sample. This figure shows a laboratory scale glucose analyzer. In 2004, Abbott Diagnostic purchased TheraSense and instruments such as the Freestyle Freedom Light Meter range produced by Abbott Diabetic Care now incorporate this wired enzyme technology. Devices of this type are highly amenable to miniaturization. Continuous measuring devices are becoming increasingly available and may well revolutionize the control of certain disease conditions. For example, with regard to diabetes, device such as Freestyle Navigator range from Abbott Diabetic Care the same wired enzyme technology as that described earlier, but now incorporate this into a tiny filament about the diameter of a thin hypodermic needle. This is inserted approximately 5 mm under the skin to measure the glucose level in the interstitial fluid that flows between the cells. The unit is designed to remain in situ for up to 5 days, during which time it can measure the glucose concentration every minute. A wireless transmitter sends the glucose reading to a separate receiver anywhere within a 30-meter range, and this can then issue an early warning alarm to alert the user to a falling or rising glucose level in time for them to take appropriate action and avoid a hypoglycemic or hyperglycemic episodes. In addition, Experimental units have also been developed that link continuous glucose biosensor measurement systems with pump capable of gradually dispensing insulin such the diabetic conditions is automatically and reliably controlled, thereby avoiding the traditional peaks and through in glucose level that occurs with conventional glucose measurement and the intermittent administration of insulin. Therefore, looking to the future, we may confidently expect to see the development of biosensor system that can continuously monitor a range of physically important analyzer and automatically dispense the required medication to alleviate the symptoms of a number of long-term chronic human illnesses. This figure shows a handheld glucose biosensor suitable for personal use.